Welcome, everybody. Thank you for taking the time to attend our webinar today. On behalf of the product development team, I'm excited to present the work that our team has done to put forth this new release, which we are calling version 3.9. I'll be going through the slide deck fairly quickly. We have a large number of slides. As Megan said, the slides are available for you to download and review at a later time. We also, in addition to the slides that I'm presenting, have added a number of other slides in the appendix section, which we won't be covering, but it has extremely useful information as a user to understand some additional features and use cases that we have also included in 3.9. With that said, let's start and review the new features that we are introducing in 3.9. So here is just a quick summary of some of the topics that we'll cover today. So the first topic, and something that we are really excited to present, is our support in a multi-cloud environment. So as you are probably aware that DRAGEN has been available on AWS for a number of years, we have now brought similar support to the Microsoft Azure platform. So we'll talk about that in a little bit. Another feature that our customers have been requesting for a while is to support variant calling for RNA samples. So that is something new that we have introduced in this release, and we'll discuss that as well. As we are expanding our portfolio in the somatic space and have introduced some biomarkers in the previous releases, in this release, we are also excited to bring in the HRD biomarker for whole genome samples. As we do with most of our releases, we are always looking to improve the accuracy of our existing pipelines. So we'll cover some accuracy improvements that we have done on the Germline side. And another application that we are investing quite a bit in so that our customers who are using our platform can also take advantage of is the single cell pipeline. And so we have some new features that we have introduced on the single cell side that we feel would be valuable to some of you. Just before we get on to 3.9, I wanted to quickly review what we had updated in 3.8. So if you remember, in 3.8, we made an introduction to our Aura compression. So we have some updates into 3.9 from there. As I mentioned earlier that, we are bringing in more biomarkers with every release. So in the previous release, we had introduced the TMB and the MSI biomarkers, and in this one, we are bringing in the HRD. We also have made updates to the HLA pipeline, and the slides are in the appendix section of the updates that we have made. So please feel free to review those once the webinar is over. And then we had also introduced the DNA amplicon feature in 3.8. So again, that's a feature that has been, getting some traction, and we have an app as well in BaseSpace that lets customers run their amplicon sample. Another update that we have made to our slide deck this time is we understand now that, DRAGEN is available in multiple platforms. However, sometimes some platforms do not bring in a certain feature, and that could be because of the way that feature is designed or, the availability at that time in a cloud or a on-site environment. So what we have done in this slide deck, just to make it easier, is share with you where a certain feature would be available. And typically what you will see is, whether we have it on premise or in our BaseSpace software as a service platform, or in our ICA platform, which is our platform as a service, which is something that we introduced earlier in the year, or if you have access to DRAGEN in your own AWS or Microsoft Azure environment, then if that feature would be available to you over there. So this is something we think would be helpful as you review the new updates that we are making in this series. Okay, so let's jump now to the new features in 3.9. So the first one we are going to talk about is DRAGEN on Microsoft Azure. So here is a quote that we worked with GeneDx on when we were designing our Azure offering, and really the push came from some of our existing on-site users, as they are expanding their, the sequencing that they do and the amount of data that they are analyzing, they've started to run to a point where they're saying that, they're spending more time managing the data, running a data center essentially versus trying to find answers for genetic diseases. So as Microsoft was a partner with GeneDx, a push came from them saying, okay, can we make DRAGEN available with Microsoft's help on Azure? So really, as our customers are scaling up, they are finding that their on-site systems are just not being able to keep up, or the costs related to that is, preventing them from going as fast as they want. So we worked with GeneDx on an early access, and they have been using our Azure platform for a couple of months already, and we've been getting great feedback from them. So thanks to GeneDx. So just an overview of what we have on Azure today with DRAGEN. Microsoft Azure has now deployed field programmable gate arrays on machines that are available in their cloud. These machines are called NP instances. So if you remember from the AWS side, they are called F1s. In the Azure world, they are called NP instances. These instances let us run in about 40 minutes DRAGEN, a sample on DRAGEN for a 50x whole human genome for small variant calling. So in about 40 minutes, you can run Mapalign, save the BAM file, and call small variants on a 50x whole human genome. So pretty fast. Still a little bit slower than our on-site, but when you can consider the power of the cloud where you can scale out, that's a pretty fast turnaround time. Today, if you want to run DRAGEN on Azure, it's available under a bring your own license model. And what that essentially means is that you can set up your environment in Microsoft Azure, and we have our application in Azure Marketplace today. And you can contact your Illumina sales rep to get access to a license that will let you run DRAGEN. And again, if you're familiar with running DRAGEN on site, the analysis, command line, and others are exactly the same. So all you need to do is, bring up an instance, have your data available in blob storage in Azure, and then you can run DRAGEN via the command line on that machine. But for now, it's a bring your own license model, and that's something that you can get via your Illumina sales rep. Just to give you an overview of the footprint of where DRAGEN is available today. So as our first wave of deploying with Microsoft, we have DRAGEN available in the 3 regions worldwide, where you have 2 regions in the US, which is East and West. We have 1 region that's available in Europe. This is in Amsterdam. And then we have 1 region in Singapore where these NP series or these FPGA machines are available. There are 3 types of machines that Microsoft has deployed. So they are really referred to as NP10, NP20, and NP40, and all of them can use DRAGEN. The difference between those 3 machines, other than, in terms of CPUs, the NP10 machine has 10 CPUs, the NP20 has 20, as the name says, and then there are differences in terms of memory, storage, and even the number of FPGA cards. So there are multiple FPGA cards in the NP20 and NP40 instances, but today in DRAGEN, we just use 1 FPGA instance across all of these. But the benefits you can get out of the larger machines are, faster speed, more storage, more memory. So depending upon your application, for example, if you're running deep sequence, tumor normal samples at 100x, 40x, then we would say run an NP20 instance because it has more memory and the analysis can run faster. If you're running whole genomes or whole exomes, maybe an NP10 instance is, or germline samples of, whole genome about 30x, then an NP10 instance may be suitable. Here is some benchmarking that we did on an NP20 instance. You can see that the small variant calling analysis, which includes the mapping and the aligning, for a 50x whole human genome takes about 40 minutes or 39 minutes here. And if you run all the callers on that sample, and when we say all the callers, it includes structural variants, CNV, CYP2D6, expansion hunter, you can get all the analysis done on a single node on an NP20 instance in about 66 minutes. So fairly fast, and you can get very good turnaround times with Azure. So again, as we said earlier, that if you have Azure access and if you want to, test DRAGEN or try DRAGEN in Azure, then please reach out to your Illumina sales rep. We also have some information in terms of running with Azure, with DRAGEN at this link over here. And again, we have also validated that DRAGEN can work with Azure Batch as well. So it has a quick start that we have developed with the Azure team, and that will let you deploy and scale with DRAGEN if you go to Azure. So again, look up our website. There's more information in terms of accessing Azure and also in terms of deploying workflows there. The next feature that I want to highlight is RNA Variant Calling. So this is the pipeline that exists today in DRAGEN. And some of you who are using the RNA pipeline with DRAGEN must be already familiar with this. But again, it's a standard workflow where you can input FASTQ files or BAM or CRAM files. You can bring in a GTF file with gene annotations. You provide the reference hash table, and you get a RNA map-aligned BAM or CRAM file. Once you have that file, you have three options which you can enable. The gene fusion detection and the quantification already existed in previous releases. And in this new release, we are bringing in the option of RNA variant calling. So the DRAGEN RNA variant calling can call directly from the RNA without needing any matched DNA samples. We support de novo RNA variant calling, so it calls variants directly, but there is also an option to do forced genotyping. So you can genotype variants from an existing variant calling file. The RNA variant calling leverages the mapper and the somatic variant calling algorithm that we have developed from our somatic pipeline to call variants from the RNA secretes. These RNA variant calling, the variants are, they don't need corresponding DNA samples, as I said above, but also you can force genotype them and you can confirm the expression of DNA somatic variants in RNA samples. So again, a lot of flexibility and a number of features that we think would be useful for anybody who wants to run RNA variant calling with DRAGEN today. On the right, we are giving you some example command lines, and in red, we have highlighted what you need to turn on for this feature. Again, the command line allows you to set the input type. So here, the input type is a FASTQ file, and you can see that we are providing the FASTQ file as tumor files because we want to enable the somatic variant caller to run this analysis. There is an option to include an annotation file if you want to, and then you can optionally force genotype with a VCF file. So again, a simple command line to turn on and turn off this feature. The RNA variant calling is about 19 times faster when we compared against the GATK, and it provides very comparable accuracy. So on the left here, we are showing you, ROC curve for SNPs and indels, and again, sensitivity versus false positives. And we have taken an HCC 1187 whole transcriptome sample, and we are just running the DRAGEN RNA variant caller versus the GATK variant caller. In the GATK analysis, we have simply taken the DRAGEN BAM and used that to call the variants. On the right, you can see the total analysis time. So if you were to run the DRAGEN RNA variant calling using the map align and the variant calling option, then it takes about 11 minutes or about 11 to 13 minutes to finish the analysis. The RNA variant calling on the GATK side by itself, takes close to 3 hours to run. So pretty fast, and we think that the accuracy is very comparable to what you might get from a GATK variant caller if you have already run that in the past. Okay. The new biomarker that we're introducing in this release is HRD. So let me just quickly, give a background on HRD if you're not familiar. So, we are aware that, the DNA is constantly being damaged and it repairs itself within our bodies. However, and again, that process is what we refer to as homologous recombination. However, when the body is unable to repair, then the double strand breaks in the DNA, and we are not able to repair those. And that is what we refer to as homologous recombination deficiencies. And previously, it was thought that this is really caused by mutations in the BRCA genes. But however, recently more genes have been identified, that cause this deficiency. But if we are aware of this condition, and if we can determine whether someone is HRD positive, then they become candidates, for certain treatments, related to PARP inhibitors. And again, and that's the reason why this biomarker is starting to, emerge, and people are interested in, finding out if somebody is HRD positive or not. So in DRAGEN, what we are doing is we are computing this genomic instability score, and it is done in your, whole genome samples today. So we are supporting this today only in whole genomes, and you can bring in either a tumor sample or a tumor normal pair. And we use the somatic CNV calling to compute the score. The workflow you can see here is fairly straightforward, where you have your tumor file, and you bring in a normal file, or you can bring in a population VCF file if you only have a tumor only sample, and we compute the HRD score and output the results in a CSV file. The HRD score is computed over these three tests, which we'll talk about in the next slide. It's an LOH score, a TAI score, and an LST score. And the final HRD score is simply a sum of these three scores. On the right, we are showing you some benchmarking that we have done against a microarray data. And you can see on the y-axis is the array data, and on the right, or on the bottom, or the x-axis is the DRAGEN data. And the results are very well aligned with the array data. And we have done this over some ovarian cancer samples. So in the past, we have identified that, in the past mutations in the BRCA samples, but now in ovarian cancer and some other genes are also contributing to HRD. So here are the three tests, or the three scores that we compute, what we refer to as the genomic instability scores, or the HRD scores. So the first one is what we call as loss of heterozygosity. And this is, again, whether you have a presence of a single allele or not. And here you can see an example where we have a large gap in one of the alleles. And the test here is if it is greater than 15 megabases or not. Another test that we run is this Telomeric Allelic imbalance test. And this basically refers to a discrepancy in the allele ratio at the end of the chromosome, as shown over here. And then a third test that we run is a large-scale state transition test. And this refers to any transition points between the abnormal and the normal regions, and the gap that is there between those regions. So here are the three tests that we run and the scores that we compute, and that determines the final HRD score. Here is an example of how this HRD score is computed. So as we said earlier, that we have to run the DRAGEN somatic CNV calling, and that is what feeds into the HRD module. And in the HRD module, we compute on a per chromosome level the major allele and the minor allele. And simply here in this figure, what you can see is in red is the major allele, and that's really the higher copy number, versus the minor allele, which is the lower copy number. And on the right, we're giving you an example of how these scores are computed. So here you can see at the bottom, we have the copy number as 0, 1, and 2. And in red, we are showing you the major and the green minor allele. And an example of how an LOH test, a TAI test, and an LST test or a score is computed. So for example, at the bottom for copy number 0, we are seeing that the LOH, or the loss of heterozygosity, is greater than 15 megabases, and that means that you have an LOH positive or a score that will add towards the HRD. And similarly for the TAI and the LST. So just a quick example of how the score is computed. The HRD command line, again, is extremely simple. So if you are running your DRAGEN-based tumor normal analysis, you could start from a BAM file, or you could start from FASTQs. all you have to do is, if it's a tumor normal sample, you have to enable HRD, and you can use the variant call allele frequency option and set this to true. And I'll talk about this option in the CNV section when we get to it. If you are running from a tumor only sample, then you can provide a allele VCF file, which is a SNP VCF file, and turn on the HRD. If you already have CNV results and you simply want to run the HRD module, then you can bring in the CNV results or CNV output files and enable HRD with that. So you don't have to run the whole pipeline if you've already run it in that. We also have advanced options in here. So as I was mentioning earlier, we have set certain defaults for those 3 scores that we compute. But however, if you want to adjust those for the LOH-CI and the LST test, then you have the option to turn them on. There is also a debug mode, and what the debug mode is essentially going to do is give you more information on how certain scores were computed or if a certain test passed or not. So that's turned off by default, but that's something you can turn on if you want to get more information. So again, it's got a lot of flexibility built into it, and at the same time, it's still fairly simple to run, and something that we think is going to be more useful as we go along, and people are exploring these newer biomarkers. Okay, let's go on to the 4th major update, which is accuracy improvement on the germline side. So there are 3 areas that we targeted in this release. So one is an improvement in the MHC region. We are now bringing in the option or the feature to improve our accuracy with references with alternate contigs by using the masking option for the alternate contigs, and an alpha feature. So this is something that we do not typically do and bring it out in the open for everybody, but we are introducing a new feature which is still in the alpha phase, which enables a machine learning aspect within DRAGEN. And I'll talk briefly about that and give you an option on how you can run that as well. So let's talk about the MHC region. So if you remember, in the summer of last year, the Precision FDA challenge that took place, there was one challenge that was related to the MHC region. And we, as DRAGEN, did not do very well in that test. And you can see here that DRAGEN 3.7, which was the release at that time, had a very large number of missed calls in the MHC region. So while we had some improvements when we had introduced the graph-based reference and we had improved SNP and indel calling, still the results were not to the point that we liked. So what we have done in the 3.9 release is we have introduced additional alternative contigs which are added across the MHC region. And really the reason to add those is it helps us to get better read mapping accuracy. There is a new BAM tag called SD that we have also included into the BAM, and that helps us to do some better read filtering to improve the variant calling specificity. And also for the HG19 and HG38, we have some selective masking that we have done in the MHC alternate contigs, and that helps us improve the VC sensitivity. So again, the changes are across the board. So we have improved the reference, we have improved how the alternate contigs are handled in the mapper, and then how that feeds into the variant caller and helps improve the accuracy through the variant calling as well. So you can see as a result of all those improvements, there is a fairly large drop in the SNP and indel false positive plus false negative rate. So again, something that if you are focused in this MHC region and you have samples that you would validate, we would be happy to get your feedback. But at least in internal testing, we are seeing this big improvement. The other improvement that we have done is how we handle the alternate contigs and its relation and the related graph-based improvements. So again, for genomes, or references with alternate contigs, in the past, we have leveraged the liftover of alignments from alternate contigs to those positions for the primary chromosomes. And that helps to boost the accuracy for sure. However, in 3.9, we now are using a masking approach for those alternate contigs. And the remaining unmasked alternate regions do serve as decoys as well. And essentially, that helps us to improve the mapping, and that trickles down into the variant calling to improve the variant calling accuracy as well. So again, this is something that's default. So when you build the hash table with 3.9, we use an internal bed file to mask those alternate contigs, and this will happen for the HG19 and the HG38 references. The native this native alt contig masking will improve the variant calling accuracy, which I'll show in the next slide. So here is a comparison. When we're using the HG002 Precision FDA sample as an example. And on the left, we are showing you the errors or the false positives and false negatives for SNPs, and on the right, it is for the indels. So here is the improvement that we are seeing. So if you were to simply run HG38 alt aware and compare it with the alt masking, you are seeing that the error rate drops. We know that going from the linear reference to the graph reference, we see an improvement. But on top of the graph reference, if you use the alternate masking approach, then we get a further benefit. So again, what we are saying is run or take advantage of the graph reference for HG19 and HG38 that DRAGEN supports. And on top of that, you get the improvements with the alt masking as well. And again, improvements are seen, if you can see on the plots on both sides for SNPs and indels. So this is moving on to this new feature called, what we are referring to as machine learning within DRAGEN. This is something, as I said, it's an alpha phase, and we would be happy to, work with you and see if you have any feedback and see if you have any results to share when you try to run this, when you upgrade to the 3.9. But what we are doing is we have integrated a machine learning, algorithm or a model to improve the quality scoring and genotyping for germline samples. This applies, to small variant calls today. It will filter out about half of the SNP false positives with smaller gains on the indel. So again, the benefit right now or the impact we are seeing is on SNP false positives, but there is improvement in indels as well. We are also seeing that the false negative counts also have reduced by about 10%. So this feature is an optional feature, but what we do is when you enable this feature, we will update the qual, GT, and the GQ fields in the VCF. There is a small penalty today, about 5 minutes, that you incur when you run this feature. But again, since this is in alpha phase, we haven't spent too much time to optimize it, and we will in future releases try to bring that time down. Today, it's available for you to run on whole genomes or whole exomes. And when you enable this feature, while you get these updated, qual and GT scores, we will still retain the original scores, and we're tagging them as dqual, dgt, and dgq. Again, you're still keeping the old scores if you want, but we also have an updated score. If you want to try this, feature, on our support page under the product section, we have the package, which has the ML model that you need to download and install onto the DRAGEN server. And when you run the ML feature, you need to point on the command line to the directory that contains the ML model. Today, again, as we said, it's our first offering of this, and it's in alpha phase. It is only supported on HG38. And hard filtering is applied at a qual 3 for this feature. And again, since this is an alpha release, we are not supporting for multi-allelic variants, for sex chromosomes, and forced genotyping yet. But again, these are all things that may come as we get to the beta and to the commercial phase. But I have some results that we were able to collect using this feature. So let me share that in the next slide. Okay, so first let me actually walk you through the workflow, and then we get to the results. So again, the analysis is fairly easy to set up. You run DRAGEN as you normally do to get to the DRAGEN variant caller. And then you turn on the ML feature, which means that DRAGEN will run VC as it normally does, and then it applies the ML model. And this is the pre-trained model and the support files that I talked to you about that are input to this ML model. And what you get at the output is a recalibrated variant calling. So fairly easy to run. And then you can run with and without this, and you can see the comparison if there's an improvement in your data sets or not. Here is our internal benchmarking. Again, we're using the HG002 high confidence truth set for this analysis. And here we are showing you on the left side the SNP and the indel accuracy. And again, the y-axis is FP plus FN. And in blue, we are showing you DRAGEN with the standard hash table and the FP plus FN. Then when we get to the graph, that's in green. So you can see, and this feature was already there for past 2 releases, where we see a big improvement using the graph reference. And then in brown, what you are seeing is the impact due to the ML model that we have. So again, we are seeing improvements on SNPs and indels, and definitely if you're using the standard hash tables, there's a big jump coming down using the graph as well as the ML model. As I mentioned on the right, we are showing you the runtime comparison. So, if you run the ML, model, you'll see about a 5-minute increase in total runtime. Again, something that we intend to optimize as we get to the next release. Okay, let's talk about the new features in single cell. So, we've been working on the single cell pipeline for the past 2 releases, and with each release, we are bringing in some additional features as we hear feedback from customers who are using that pipeline. So there are 3 features that we have done in this new release. Is there in 3.8 we had genotype-based demultiplexing. We have simplified that into this genotype-free demultiplexing. There is an option to do feature counting, and also a new technique in terms of how you cluster cells and how you identify cells from multi-samples with that you have demultiplexed in your experiments by using the cell hashing approach. So again, coming to the first feature is the genotype-free demultiplexing. So what the genotype-free demultiplexing does, it can demultiplex your scRNA samples without needing any sample-specific genotype data. So with this method, there is no need to input the sample's genotypes. However, instead, what you can do is you can input a reference genotype file from a genetically similar cohort. So you do not have to have VCF files using the genotype information that are individual to a sample, but you can have a cohort file that is similar to that. And that makes the analysis steps much more easier and faster for you to run. So again, what we do essentially is we cluster the cells, and the genotype probabilities are then computed for each cluster. And probabilistically, we assign these clusters in a genotype-based sample demultiplexing. So it just simplifies the process and helps to demultiplex these RNA samples without needing to provide a sample-specific genotype file. Again, what you can do is you provide this reference VCF, what we have highlighted in red, and you indicate the number of samples that you have in your experiment so that we can identify the total number and use them for clustering. Another feature that is emerging or another aspect in single cell RNA is feature counting, and that is again something that is an emerging area. So we are now introducing that in DRAGEN, and what it essentially does is it measures the expression of cell surface proteins or antibodies along with the expression of genes on the same cell. And this feature is now available in DRAGEN, and it is fairly flexible, where you can bring in various read configurations that you have in your experiment, but also it has support for UMIs as well. So again, something that we see as an emerging feature in the single cell world, and something that one of our customers had approached to us and said if we could include this in DRAGEN. So that's available today, and what you need to do in this case is you need to bring in a reference file, and then you indicate that these the feature that you want to enable for this measurement is for antibody measurement. So again, in red, we are enabling or showing you what the command line options are. cell hashing. So the cell hashing, as I was saying, is a new technique where you can assign sample identities to cells in a single cell RNA experiment. Again, what is done is it uses an oligo sequence to tag a part of the read, and those oligo sequences are then used to identify the various cells that you have in your single cell experiment. So again, in this feature, on the right, what we're showing you is you need to provide us as an input the cell hashing reference or these oligo sequences or these these tags that we need to use for the cell hashing experiment, and then you let DRAGEN know that this file is equivalent for the cell hashing feature, and then we can use that to demultiplex the cell. Here is again, the single cell workflow. So you can bring in your RNA FASTQ files or your protein FASTQ files, and then you map align, or you can even start from the BAM files, and then you have the option to turn on all these features. So you can in the RNA pipeline, we do cell barcode and UMI error correction and read counting, and then in the boxes that have the stars in them, those are the optional files that you can include in your experiment. So either it could be the feature reference file or the cell hashing reference file or the genotype-free demultiplexing DCF file. And what you get as an output from the single cell pipeline is a cell by gene expression matrix, essentially. And that file then can be used downstream in a number of tools like Surat or other tools that you want to use for your downstream experiment. Okay. Now we're going to talk about all the updates that we have made to our existing pipelines. So what we have here is we will talk a little bit about the update that we have done on the CNV pipeline. So there is now, if you are running tumor normal CNV pipeline with small variant calling, then we now have an improved workflow where you can do this in a single execution. So in the past, when you were to run somatic CNV calling, we required the matched normal to first go through the germline SNV caller. And what that meant was you are now doing an additional run. Now in this 3.9 release, when running with the somatic SNV caller in tumor normal mode, the matched normal heterozygous sites can be leveraged directly within the somatic CNV caller in the same DRAGEN execution. Additionally, somatic SV can also be added in the same execution. So what we are saying is that you can run whole genome tumor normal analysis as a single execution, and you can call small variant CNV and SV, and that simplifies your existing your entire workflow versus previously you had to run the germline caller separately as well. With the simplified somatic workflow, what we are showing you on the left is the accuracy comparison versus running the previous way of doing things and now what we support. So prior to 3.9 or 3.8 and earlier, you had to provide the CNV normal B allele VCF file. And now in the 3.9 release, you have the option to use the CNV somatic VCBAF file. So this is the file that is internally generated within DRAGEN when you run the somatic caller versus what you had to provide. And you can see that the results are very similar to each other. And on the right, what we are showing you is the runtime improvements. So on the top, we are running the tumor normal analysis for whole genome in a single execution, versus at the bottom, we are showing you that the execution requires you in green to run a normal sample as well. And so not only is your execution now simpler, but also it runs much faster when you compare with what you had in 3.8 or before. So again, just an improvement to make it easier for you to run whole genome tumor normal samples. We have also updated, the somatic CNV calling for enrichment applications. And again, this applies to essentially whole exomes and targeted panels. And what we have done is we have leveraged the panel of normals to estimate systematic biases in the library prep. And with that, we also have improved the somatic calling and the scoring model for tumor inputs. You can still bring in a CNV segmentation bedfat for gene level analysis, and the CNV events are reported as copy ratios with respect to the panel of normals. So again, this was an area where we felt our accuracy was not up to the mark, or at least as what we expected for somatic enrichment samples. So those have now been updated in this release. A brief update on the structural variant calling. There are three improvements or updates that we have made in the structural variant calling. One is we introduced this liquid tumor mode, and I'll talk about it in the next slide. There are two other features that we won't cover in the webinar today, but we have slides to show the improvements for these in the appendix section. So we have, as you can remember, if you remember in 3.8, we introduced improvement in the FLT3 ITD detection, and now there is an improvement on that. And also there is an improvement if you use the forced genotyping option in the SV caller. So specific to the liquid tumor mode, so liquid tumor mode basically helps to restore some SV recall for somatic analysis when you have contaminated normals. So the liquid tumor mode has a mode in the SV calling that adjusts the tumor normal somatic calling module or the model for the tumor in normal contamination. So when liquid tumor mode is enabled, SV recall can be substantially improved if you have high tumor in normal contamination. Today, the liquid tumor mode must be enabled. So if you have liquid tumor samples and if you are aware of contamination in those samples, then you can enable this mode. The default mode or the default level is 15%, but it can be adjusted if you have more contaminated samples. So again, the command line to enable this is fairly straightforward. You turn on this feature by setting the liquid tumor mode to true. And if you want to adjust the tumor in normal contamination tolerance, then you can set it with this option as well. So we are giving an example. If it's 25%, the default level is set to 15%. Here is some benchmarking data that we have. So this one is a tumor in normal contamination simulated sample. We mixed two germline samples with high quality truth sets, so NA1278 and NA24385, and we simulated with an 80x tumor coverage and a 40x normal coverage, and then we tested with samples which have 0% contamination and then samples with 15% contamination. So if you were to use the default SV with 0% contamination, then you get the recall and the precision at this level. If you were to turn on or to bring in the sample with 15% contamination, but you did not turn on the tumor, the model for liquid tumor contamination, then you can see that the recall substantially drops. It becomes really bad. However, when you turn on the liquid tumor mode, then we account for that contamination, and then the model for this tumor mode, liquid tumor mode kicks in, and then you can recover a lot of the loss that you had if you didn't have that model. So again, something as we think is important as we see more and more applications with liquid tumor samples. Okay, let's go to the RNA Gen Fusion section, and what we're introducing in 3.9 is better gene fusion detection in repetitive regions. So, if you're aware, there are clinically relevant fusions, such as, what we show here as the DUX4 and the Septin14. And again, these are challenging with one or both breakpoints in the repetitive regions. And these are challenging to detect with the standard gene fusion calling algorithm that we have. So in 3.9, we have implemented a targeted alignment approach to report fusions in these repeats of interest. So by default, internally within DRAGEN, we will automatically apply, this to these target regions, as I said, the DUX4, the IC, Septin14 for HG19 and HG38. However, if you want to bring in, certain genes or repeat intervals, that are of interest, then we have an option, as we are showing here on the right, to bring in either those repeat genes or those repeat intervals. But again, something that we will still focus on in the future release is to bring in more support for these repeat regions, because these are clinically relevant and of interest to many of our customers. So this is our first improvement that we have made. And in the next slide, actually in the appendix, we moved some of the results, so you can see some of the results we have in those regions. The next feature that I want to quickly talk about is the improved annotation for RNA fusions. So if you are aware, we've introduced Nirvana for the past couple of releases in DRAGEN, and in 3.8 we were able to run it end to end. Now we are making some improvements within Nirvana to have improved HGVS R notation for gene fusions essentially. So previously our formats were not well aligned with HGVS, especially for RNA fusion calls. So we have made that improvement in this release. And the big improvement that we want to highlight here is additional somatic and germline data sources. So fusion catcher gene fusions including 13 germline fusion sources, 22 somatic sources, and 3 oncogene sources as well. And also cosmic cancer gene fusions are included in here. So improvement that we made on the HGVS RNA notations, improved the format, better notations for in-frame gene fusions, out-of-frame gene fusions, but also bringing in more sources for fusions for annotation that we did not have in previous releases. Just a quick set of updates on the somatic small variant caller in this release. So one feature or one request that we were getting from customers is to enable GVCF capability for somatic pipeline. So if you're aware, for the germline samples, we do support this, but we did not have this feature for a somatic pipeline. So again, in contrast to the regular VCF, a GVCF file will cover all the positions in the genome, right? So what we have done is that when you enable this option to generate GVCF somatic, we will bring in an artificial on-ref tag that is used to represent these variants. And what happens is with this feature, the output can be then used to gain confidence in the absence of a variant. So you can turn this on. By default, the GVCF output is compressed. So, you can get a smaller GVCF file. But you do have an option if you want to do base pair resolution as well as we do in the germline sample. But then again, that will come at the cost of size of the file as well as the time to run the analysis. But the outputs are similar to what you would get in the germline side, and you would in the somatic side you would get a hard filtered GVCF file as we do on the germline side. So very similar, but it just lets you check for any missing variants and see what was the cost for that and why did DRAGEN not call it in the VCF mode. There is also an improvement that we have done in mito calling. So in the past, we have been slowly updating our somatic variant calling algorithm from a mutex 2-like approach to a Strelka 2-like genotyping algorithm. The mitochondrial calling within the germline analysis does use the somatic calling, but that was not updated till now. So now we have made that update, so the mitochondrial variant calls that you would see will now start to report a score in the format of an SQ or a somatic quality versus an LOD that was done in the past. Also, multi-allelic calls are now possible with the mito calling from this release. We are also able to target allele frequencies down to 1% when the coverage permits. Again, we are showing you on the right some results that we have aggregated over 10 different samples. And the big improvement we are seeing is in the precision for mito calling. So if you see, the sensitivity was already improved in some updates that we had made in 3.8, but however, the precision was lacking quite a bit. And you can see that in 3.9, there is a big jump in mito calling precision. On the right, we are showing you as well the total error counts as we progress from using Strelka by itself to DRAGEN 3.8 and moving to DRAGEN 3.9. The big update or the improvement is in the false positive rate, as you can see here. So again, I know a number of you are using DRAGEN for mito calling, but and so I think this update will definitely benefit quite a few of you. I want to quickly go over some of the Aura compression updates that we have made in this release as well. So there are 4 different updates that we have made. So one is a compression of paired read options. We have sped up the compression by using the hardware within DRAGEN. If you're using Aura compressed files in the cloud, we now support streaming, and also the option to go directly from DCL to FASTQ. So let's go one by one into a little bit of more detail for each of these features. So in DRAGEN 3.9, now we have an option to interleave, to compress interleaved pair of read files. So if you have read 1 and read 2, you can provide those as inputs, and we can compress a pair of files together, and what you get is a single interleaved compressed Aura file. And what that means is you get one file for a pair of samples. That file is also further compressed by about 10%. So if you were to individually compress those 2 files versus interleaving those files and compressing it as a single file, you get further 10% file reduction. And this interleaved read file can be used as an input to the DRAGEN mapper as well. So you do not have to decompress it or de-interleave the file to bring it back into a pair and input that into DRAGEN. You can provide an interleaved input file to DRAGEN directly, and internally we will take care of getting the read 1 and read 2 files out of there. So again, something that I think will simplify on how you manage the files, but also give you further file reduction and makes it simpler to run within DRAGEN. We are also using the hardware unzipper within DRAGEN on the FPGA. So there is faster compression of the FASTQ.gz file. So again, something that we didn't have in 3.8 as our first offering of Aura, as we are looking at areas in which we can optimize and make it faster to compress, decompress, we are bringing in those features. We are also supporting now streaming. So if you have compressed Aura files in AWS or in Azure, we can now stream those files when you run DRAGEN in the cloud directly from either the S3 buckets or the blob storage. So again, you don't have to copy those files locally as you had to do in 3.8. Now you can stream them directly, and just makes it more efficient to run DRAGEN in the cloud, saves you cost by not having to copy files back and forth. And then another feature that I think will be very useful if you are starting to explore the Aura option is, in the previous release, if you had to get Aura files from DCL, you had to do it in 2 steps. You had to go from DCL to FASTQ.gz, and then FASTQ.gz files get compressed to Aura. With this release, you can go directly from doing DCL conversion and generating your FASTQ.aura file at the end. So again, just simplifying the whole end-to-end workflow if you are using this Aura compression option in DRAGEN. I want to also highlight a major update that we have made for the Sna caller in DRAGEN. So as you are aware that an SMA is a neuromuscular disorder, and it affects a fairly large number, 1 in about 6,000 to 10,000 live birds. And there is also a carrier frequency of about 1:40 to 80, depending on the ethnic group that you're coming in. So the DRAGEN SMN caller now is capable of identifying, the SMN1, SMN2 copy numbers from whole genome data. But also in this release, we are bringing in the option to provide carrier status for that. So the SMA and the carrier status is reported with the SMN1 and SMN2 copy numbers. You can see that again today it's available for whole genome data, and you can simply turn it on by using the enable SMN equal to true command line option. It's available for these 3 references. So these are typically the 3 references that you would use. And then you get an output file that gives you whether your SMA is true or false. It tells you your carrier status, and it also tells you the copy number for SMN1 and SMN2. This is the workflow that we have. So again, you do map-align, with DRAGEN, and then you have the algorithm, to compute the SMN copy number, and it tells you the SMN copy number at 8 different sites between SMN1 and SMN2, and then it lets you know what your SMA and the carrier status is. And depending on this flow over here, it will indicate whether you're positive or not and whether you're a carrier. And again, the requirement today is the sample has to be whole genome, and it has to have sufficient coverage with at least 30x. So here is some benchmarking we have done. So we have validated against 250 samples, clinical and from the 1,000 Genomes project. There were 42 samples that were SMA affected and 23 samples that were known to be carriers of SMA. So this is an orthogonal validation also that we have done with a digital PCR or MLPA. And here you can see that the results for SMA and the carrier were concordant. And we are reporting, the SMN1 carrier, the copy number, as well as the SMN2 copy number as well. So again, highly concordant results, an improved SMA algorithm, plus giving you carrier status information which did not exist in previous series. We are looking another feature that we are bringing in 3.9 is more options for disomy detection. So the first one that we are bringing in is UPD. Again, UPD is this is our initial release of this feature, and today the detection is I would say limited to whole chromosome UPD. We are looking at giving you subchromosomal UPD and further detection modes in future releases. But in this first release, this is something that we are leveraging our the output of our SNB caller, and we are measuring the het-home ratios at these SNB sites. And what we are outputting from the small variant caller is a per contig het-home ratio, and depending on that het-home ratio, we can determine if there is whole chromosome UPD, and that is detected, or a disomy is detected if that het-home ratio is close to 0, and if it's not there, or the normal ranges that we expect would be between 1.0 and 2.0. Again, so this is something that we've already output, but now we are leveraging this information to give you, the disomy status for UPD essentially. So again, something that we're bringing in, we're looking at other disomy or trisomy, detection methods and algorithms as well, and again, expanding this in future releases to be subchromosomal as well. A quick review of some of the methylation updates that we have made in this release as well. So, there are 4 updates that we are making for methylation. We are using, a better, we have improved the usage of the alt-aware reference and the hash table options that I was talking about in, the previous sections. We now have a strand-aware dedupe option for better alignment. And also, we are compressing the methylation report, so that way the final output is a much smaller file than what we were doing before. And again, we are improved, the output metrics. There is better mapping metrics for multi-pass mode, better mapping metrics for single-pass mode. So if you remember in 3.8, we improved the analysis of methylation samples in the single-pass mode for speed. So, that there is some better metrics that we are putting out in this release. And that's something that we have as slides in the appendix. So just quickly going through this, we now have improved hash tables that you can use for methylation. So, you can set the HT methylated option to be true for multi-pass mode. And then if you say a combined hash table, to be set to true, then you can use that for a single pass mode. And like the multi-pass mode, the single pass mode is now fully alt-aware supported. So again, something that we want to push our users to is to get to the single pass mode, and we're bringing in all the capability that we previously had in the multi-pass mode. Again, we are now improved, how we're doing the deduping. So previously we were strand agnostic, but now we are strand aware when we're doing the dedupe option. And as a result of that, you can see that, we are getting, a better correlation, and also matches better with what Bismarck offers from a strand-aware point of view. Again, just the speed and runtime, of DRAGEN, you can do a typical, whole genome analysis, if you, have the compressed files versus the uncompressed files as an output. Extremely fast. If you get the compressed file, you get much smaller files, about 5x reduction, but there's a small penalty in terms of the runtime that you get out of these out of the cytosine report files that DRAGEN outputs, either if you're doing a single pass or a multi-pass option. Just a quick overview of all the base app updates that are going to happen with 3.9. So what we're going to do is we're going to bring in, expanded set of reference hash tables. As we were showing above, we have the HG38 and the HG19 samples, which are in graph genome. So we are going to bring in that. We are going to bring in that into the somatic applications as well. We will be bringing in all the biomarkers that I talked about earlier. We will also enable the HLA calling, which was not done from the 3.8, but we bring into 3.9. And the usability improvements, for the somatic CNV calling that I mentioned about, that you can run everything end-to-end as a single workflow for whole genome tumor novel samples. Again, we have 3 core apps in base space, the germline, the enrichment, and the somatic side. And this slide kind of shows you the new features that we'll be bringing in 3.9. So the HLA calling will go across the board. For the somatic analysis and enrichment and the somatic pipeline, we're bringing in the biomarkers. We're bringing in the new graph reference genomes with, alt masking capability for the germline and the somatic applications. The somatic GVCF outputs will go to the applications that support the somatic data. And then on the germline pipeline, we're bringing in the new SMA caller that I talked about earlier. Again, for everybody on the call, if you want to learn more about what's in DRAGEN 3.9, you can download the software and check our user guide that is available on our support page as well. And that was a quick rundown of all the features. There is more in the slides in the appendix section that you will have access to, but I would like to stop here and now address some of the questions that you may have from listening to this talk.